Now. So today we'll be going over glycogen metabolism and pentose phosphate pathway. So glycogen, it's basically a stored polymer form of glucose. So whenever the cell has a lot of glucose, it's an efficient way for it to store it for later use. So whenever there is a lot of like the cell is in a high energy state, it will want to store glucose into glycogen. And the benefit of that is when the cell needs energy, needs glucose, you can break down glycogen and you get really fast glucose right away. So this is kind of an overview here of glycogen being stored. So we have G6P here down at the bottom. So you would go from glucose to G6P, and then that's converted to glucose 1-phosphate, and then eventually that becomes glycogen. And then to go the other way, we break it down. So this is just a general overview. Okay.
Do you have
So
these notes in GoodNotes?
what?
Do you have these notes in GoodNotes?
Yeah, yeah. Is it better in GoodNotes or?
No, I was just wondering if this was in GoodNotes or if you could like send it to me so I can like write on it while you talk.
No, yeah, it is in GoodNotes. If I are you gonna write it down as I'm going?
Well, I'm writing it down too, but I just figured then I could write what you say and like highlight stuff.
Okay, okay. So I'll go a little bit slower so you have more time to write stuff down. Okay.
Okay.
So the slide I had in the beginning, we'll go over it again in more detail, so you don't have to write that down. Okay.
Perfect.
So this is storage of glycogen. So gluconeogenesis. So that's making glucose back. That's mainly done by the liver and the kidney, and that's what we went over last time. So glycogen storage, that's basically where glycogen will be stored. Mainly this happens in the muscle, the brain, and then the liver actually does some glycogen storage as well. So yeah, it's mainly stored in the muscle and the brain. So in the brain, it's stored in astrocytes, and it's stored as a polymer of glucose. So cells can either use fatty acids to store energy, or they can use glycogen. So the benefit of using glycogen is it's easier to control, mainly because fatty acids are hydrophobic, and you won't be able to hydrolyze them with water just as efficiently as you can with glycogen. So glycogen gives you immediate access to glucose uh, over fatty acids, I guess. So when glycogen, you're able to break it down faster, and you're able to use glucose right away. And then regulation of glycogen metabolism and breakdown, it's generally organ specific, so it depends on where we are. And then it can also be affected by hormones as well. Are you good with this slide, Ali?
Yeah, I'm good. Thanks.
Okay. So this is glycogen versus amylose. So amylose is used by plants, and I kind of just mentioned this to explain why mainly mammal cells use glycogen. So glycogen, which we'll talk about, has linear forms and branches, and the branches allows it to be more open, and then this allows immediate use of it right away. The branches actually excludes water, and this allows for the degradative enzymes to produce the glucose molecules right away. When if we use amylose, which is the linear form, you won't be able to. There's not water around interacting with the amylose to allow for the breakdown of amylose efficiently. So glycogen allows us to use water and break it down more efficiently over amylose. And then this is a picture of like glycogen, of just the general idea of it, and we'll talk about how it's synthesized and everything. Okay, so here I have something called Lafora disease. This is basically a disease where you have insoluble glycogen. So the deficiency is in a gene called EPM2A, and it produces the protein laforin. Luforin regulates the covalent remodeling of glycogen. So whenever you have a deficiency in this gene, you have a deficiency in luforin, and glycogen won't be remodeled. And this actually allows the glycogen to not be broken down, so it starts to accumulate, and then you have this what is called Lafora disease, and then this can result in severe epilepsy, and then it can even get worse if it's not treated early on. This disease actually uh, allows us, pretty much tells us that glycogen is remodeled, and it's important for the storage of it, because whenever we have a deficiency in the enzyme that does the remodeling, glycogen is insoluble and it can't be broken down. Okay, so this is the general structure of glycogen. So you have these glucose monomers, and they're linked with alpha 1 for linear linkages. So the alpha is, so this carbon over here, the anomeric carbon, and it's linked with the, so alpha just means the oxygen atom is facing down, and 1 4 is just means that it's linking carbon 1 and carbon 4. So those are the linear forms of glycogen, and it also has alpha 1 6. So alpha just means the oxygen atom here is pointing down, and it's linking glucose monomer at carbon 1 and another glucose monomer at carbon 6. So it's synthesized via two enzymes, and it's broken down with two different enzymes that kind of do the opposite of what the synthesis does. So the linear linkages, which are the alpha 1 4, those are catalyzed by an enzyme called glycogen synthase. So the glycogen synthase just links the alpha 1 4s. And there's another enzyme called branching enzyme, and this enzyme catalyzes the branching. So this catalyzes the alpha 1 6, and it allows glycogen to be more branched. Glycogen is then, when we need to break it down, there's two different enzymes that break down glycogen. It's one of the enzymes is called glycogen phosphorylase, and that one removes the alpha 1 4's units. So this one kind of does the opposite of what glycogen synthase does. And then the other enzyme we have is the branching enzymes. So this one allows us to take care of the alpha 1 6, but it doesn't break it right away. What it does is it removes the alpha 1 6 unit to an alpha 1 4 position, and then it allows glycogen phosphorylase to break it down. We'll go in more detail with what all these enzymes do and some of the mechanism for some of them, but just not confused, the branching enzyme just moves the alpha 1 6 linkage, this one right here, into a non-reducing end, and then it allows glycogen phosphorylase breaks it down. Okay, so glycogen can have reducing ends and non-reducing ends. So if we go back here, whenever we have, so these are called anomeric carbons, which for glucose it's carbon number 1. If this carbon is not bonded to something else, it's called the non-reducing end. So this carbon over here has a alpha 1 4 bond, and then this one down here also has the alpha 1 4 bond. So those are called the non-reducing ends because the anomeric carbon is participating in a reaction. The reducing end of glycogen tends to have a free anomeric carbon. So this carbon number 1 over here is not participating in any bonds, and it's basically called the reducing end, and it allows the glycogen to act as a reducing sugar. And we'll see those become more important on how glycogen synthase works, like the mechanism of it. So we'll see here that reducing ends are actually where, so this is more of a general overview of like the glycogen molecule. So the reducing ends is where glycogen synthase starts, and then it starts to branch. And whenever you add more glucose monomers, they're added to the non-reducing end. So we usually have one reducing end because that's where glycogen starts, and then we will have multiple non-reducing ends. Okay. So this is a general overview of how this is the same slide I had at the beginning of how glycogen is formed. So usually we start it starts with glucose 6 phosphate, and this can come from many different sources. One way that we generate G6P, glucose 6 phosphate, is from glucose. So glucose would be converted to glucose 6 phosphate by hexokinase. So glucose 6 phosphate can then enter glycolysis. If we need more, if the cell needs energy, so we would run glycolysis, we need energy. And if we have a lot of energy, we'll store this G6P into glycogen. So if we have a lot of energy, the first reaction we go through is phosphoglucomutase, which takes glucose 6 phosphate and converts it into glucose 1 phosphate. And then we add a UDP to glucose. I'll go over this in more details, but we just activate the glucose, we add UDP to it, and then glycogen synthase takes UDP glucose and adds it into different adds it into like the polymer form, which you get glycogen. And this is done by glycogen synthase, and branching enzyme adds the branches. The branching and phosphorylase just do the opposite of that, and we get glucose 1 phosphate back. Okay, so let's look at UDP first, and why we use UDP when we're storing glucose as glycogen. So UDP is added to glucose, and this reaction is irreversible. So as soon as we add UDP to glucose, we're kind of committed to storing glucose as glycogen. So if we go back here and we see G6P go into G1P, these two substrates can enter different pathways in the cell. As soon as we convert the UDP glucose, we're kind of committed to storing glucose as glycogen. So another reason why we use UDP as well is because it's also a good leaving group, and it also tags the glucose molecule for biosynthetic reactions. So as soon as UDP is tagged to glucose, it kind of tells the cell that you're going to use that glucose to synthesize glycogen. And it plays a role in the catalytic activity of the reaction. Okay, so let's look at the first couple of reactions. So this is G6P, glucose, and it has a phosphate on carbon 6, and then we convert that to glucose 1 phosphate. This is done by phosphoglucomutase. So this is the same thing over here. So we just take G6P, convert it to G1P. And then after we have the G1P, this is done by the enzyme, let's go back, UDP glucose pyrophosphorylase. So this will take the G1P, adds UTP, and then we'll get UDP in glucose. So the phosphate on G1P over here attacks the alpha phosphate of UTP, and then you release the pyrophosphate, which is 2 phosphates, and then you end up with this uridine, and then you have the diphosphate, which is DP, and it's also linked to glucose. So this over here is actually not what makes it the reaction irreversible. The pyrophosphate that we generate is cleaved by a pyrophosphatase, and this cleavage of pyrophosphate is coupled with the UDP glucose formation, and this is kind of what drives the reaction forward and allows this reaction to be irreversible. So once we make the UDP glucose, we can start storing it as a polymer. Because this reaction is irreversible, that means UDP glucose cannot go anywhere except for glycogen, so we can start storing it. So the enzyme that does that is glycogen synthase. So this is what adds the alpha-1,4 glucose molecules together, and it extends the chain. So it has different isoforms. So it's different isoforms in the liver, the muscle, and the brain as well. It's a tetramer. That means it's made of 4 subunits, and then The what we'll see is the mammalian versions of this are regulated by allosteric and phosphorylation, and this depends on the tissue that we're in. So the regulation of the enzyme is dependent on where exactly are we are in the body. Okay. Ali, am I going too fast or should I slow down?
Sorry, no, that's good. I have a question though.
Yeah, go, go, go.
I was writing stuff aside. You said it, I just missed it. And
Mhm.
on this slide, it said liver and muscle isoforms. Is that what exactly does that mean?
Liver and muscle isoforms here.
Yeah, what does that mean?
Oh, so so once the gene is transcribed and then translated, uh, remember like different exons can come together and make different proteins, like alternative splicing. You can make different kinds of proteins from the same gene. So the liver and the muscle use different isoforms, which means different proteins from the same gene that have different alternative splicing. So it's generally the same enzyme, but it has different like exons that are being linked together. Does that make sense?
Yeah, that makes a lot of sense. Thank
Yeah,
you.
yeah. So isoforms is just different alternative splicing of a gene, and it gives you different proteins that are in the same family. They're just called isoforms. Okay. So now that we made G6B and we're ready to add it with glycogen synthase, let's look at the mechanism of glycogen synthase. So there's a different protein called glycogenin, and it kind of acts as a primer of where we'll start glycogen synthase. So this protein over here is what tells the cell is the starting point of every glycogen polymer. So this glycogenin has a tyrosine residue at number 194, and so this is the OH of tyrosine. And what happens is this OH attacks UDP glucose. The UDP is released, and then you kind of get this glycogenin and then glucose. And then this reaction is repeated again with carbon number 4 instead of the tyrosine residue, and it attacks another UDP glucose, and you kind of go on and keep adding different glucose monomers. So if we look here, so this is the reducing end. So this is carbon number 1. This always remains attached to our starting point, the glycogen. And then every other glucose monomer that we add, we're adding it to carbon number 4, which is the non-reducing end. Kind of helps you track of where the different glucose monomers are being added. So they're added from their non-reducing end to the reducing end of the new UDP glucose. Okay, let's look more at the specific mechanism of glycogen synthase. So what it does is it cleaves UDP and it stabilizes it by the enzyme, and then glucose is then transferred to the non-reducing end. So this is pretty much just summarizes everything I said in the other slide. So we have UDP glucose here. This is the glycogenin, and you would have glucose here. And then it would attack this UDP. UDP is released, and then you would keep adding different glucose monomers. The UDP that's released can participate in this reaction down here. It would react with ATP, and it would generate UTP and ADP. So to run glycogen synthase, we have to make sure that we have a lot of ATP. So the cell needs to be in a really high energy state because this ATP is needed to recycle the UDP back to be used in to be used to activate glucose monomers when we're ready to add them as a polymer. So after we make a couple of residues, so this is glycogenin over here. This is a reducing end, and we keep adding different glucose monomers down here to the non-reducing end. Once we get to around 11 residues or so, we have a different enzyme that comes in. So The enzyme is called glycogen branching enzyme. What this enzyme does is it takes around six residues from the non-reducing end, and it moves them to a carbon number 6 on an interior position. So this is glycogen over here. These are the six residues, and what it does is it takes these six residues and it moves them into an interior position, and it creates this alpha 1,4 linkage, which allows glycogen to be branched. And then once you create this branch, you can continue running. You can continue with glycogen synthase again, extending the chain. And then when you get around 11 residues or so from the branch point, glycogen branching enzyme comes in and adds the glucose to an interior position. And it's kind of repeated to make, and you keep doing that to create this polymer of glucose that's really branched. Does that make sense? Do you have any questions on here? This is, yep, this is supposed to say interior. Let me fix
No,
it.
I caught that.
Yeah, I'm gonna fix it real quick. Interior. Okay, so this is how everything we went over is kind of like how glycogen is formed. This now we'll go over on how it's broken down. So to break it down, you have two different enzymes that come into play. So you have a phosphorylase and a debranching enzyme. So most enzyme actually when they break carbohydrates, they use water, and that they're called hydrolases. However, glycogen is broken down differently. It's broken down by a phosphorylase, and a phosphorylase works by breaking down different residues, but it uses free phosphate. So what phosphorylase does is it uses this free phosphate, it adds it into a glucose. You get this glucose 1-phosphate, and it releases them. So it kind of just goes in and releases these glucose molecules as glucose 1-phosphates. The it's important. It's advantageous for the cell to use phosphorylase to break down glucose because we generate G1P. G1P can be converted to G6P right away using. Let's go back. So G1P can be converted to G6P right away using phosphoglucomutase. So this reaction is reversible. So we can convert it right away to G6P, and then G6P can enter glycolysis right away. We kind of skip a step when we generate G1P right away. We skip the step from glucose to glucose 6-phosphate if we were to run glycolysis normally, and this kind of saves the cell an ATP when it does it this way. So I'll go back to the other side. Okay, so when we generate G1P, we can kind of enter glycolysis, and we don't have to spend the extra ATP that we usually do in the first step of glycolysis. So phosphorylase continues to remove glucose molecules until it gets to around 4 residues from the branch point right here, and then it can't go anymore. The enzyme is generally too wide to be able to fit in and continue to break these. So then we have a different enzymes that comes in and helps us remove the other part of the inside of the glucose chain. So this is done by the branching enzyme, which we'll talk about in a second. I just kind of right now I want to go over the glycogen phosphorylase mechanism real quick before we talk about the branching enzyme. So it uses PLB as a coenzyme. Let's see, hide this. Let me go back. I don't know what I did. Okay, I'm back. It uses PLB as a coenzyme. PLB is just vitamin B6. And let's go down here. So this down here, this is the glycogen that we're trying to break down. This is the enzyme, the glycogen phosphorylase, we have PLB right here, so PL, and then this is the extra phosphate. And then this down here is the free phosphate that we use when we're breaking this down. This is, so this right here, the picture I have here is the PLB, or vitamin B6, which is, it's reduced in this picture to just PL and then phosphate. And this phosphate is important because it acts as an acid. So the way the mechanism of this enzyme works is you have this enzyme, and you have PLB, and you have a phosphate and the glycogen complex. The first thing it does is it makes a complex out of all these four things. After that, the PLB, which is down here, it gives a hydrogen to phosphate. So phosphate over here grabs the hydrogen from PLB, and then the glycogen grabs hydrogen from phosphate, and then you make this oxenium intermediate. So this oxenium intermediate over here, after it grabs hydrogen from the phosphate, so this is what I meant by the phosphate acting as an acid. It gives a hydrogen to the glycogen complex, and you make this oxenium ion, which is then reacts with phosphate over here. So we'll see that the phosphate acts as a nucleophile, attacks this double bond that has a not the double bond, this oxygen over here that has the plus sign, and at the number 1 position, and then you create this G1P, which is released. So the G1P is released, and then PLB is released with the enzyme, and it has the hydrogen, which can then start the process again to release more G1Ps. Okay, so that's kind of how we're breaking down these different glucose molecules. Next, like I said, once we get the 4 residues, we need the branching enzyme to come in and help us cleave the rest of these. So the branching enzyme actually has 2 different reactions that it catalyzes. The first thing is it transfers these 3 glucose molecules from the original branch we were breaking into the branch that's like it's linked to. So it has a transverse activity. It shifts these 3 glucose molecules down here, and once it does that, we're left with 1 glucose molecule that's linked with an alpha 6 linkage to this original chain, and then the hydrolase comes in, and because it has 2 different mechanisms, so it has the transferase and the hydrolase. The hydrolase then uses water to cleave the last glucose that we have. So what we see, the phosphorylase releases glucose 1-phosphate. So this thing over here that we talked about, so that every time it runs, it will release a glucose 1-phosphate. And the branching enzyme, when that runs, it releases free glucose. So those are kind of important with labeling molecule. Just know that the branching enzyme releases free glucose, and then phosphorylase usually releases G1P. Okay, so after we release G1P, phosphoglucomutase, it's a reversible reaction. G1P can be converted to G6P, and then G6P can be run into glycolysis in muscle cells. So whenever muscle cells are trying to break down glycogen, they usually do so because they need the energy right away. They need the energy to contract. So then they would break down glycogen, and then they convert G1P to G6P, which is then fed into glycolysis right away. So the muscle usually degrades glycogen when it needs energy. The liver, whenever it degrades glycogen, it does it for a different reason. So whenever the liver degrades glycogen, it's because we have low blood glucose levels. So when the liver degrades glycogen, we get G1P, and then we convert it to G6P. G6P is then dephosphorylated, so we get normal glucose, and then it releases that free glucose into the blood. So whenever the liver breaks down free glycogen, it's mainly to release glucose into the blood to increase blood glucose levels. And I also added that all of glycogen synthase and glycogen synthase and breakdown both occur in the cytoplasm. Lysosome can also do some glycogen degradation, but you guys don't have to know much about that. Okay, so that's kind of the everything about glycogen synthase and breakdown. I'll go over. I have the regulations as well, but the free energy of synthesizing glucose and breaking down are both negative, so they're both favorable pathways. Depending on whenever the cell needs to do it, it's a favorable pathway, but it can be regulated with other things as well. Okay, so regulation of it is dependent on regulation of glycogen breakdown and synthesis is dependent on the tissue that we're in. So in muscles, G6B can activate synthase, and AMP. So synthase would work to make glycogen, and then phosphorylase works to break down glycogen. So whenever we have a lot of G6B, that means we have a lot of glucose, we have a lot of energy. We're gonna try to store the glucose into the branch form as a polymer. So that's why whenever we have a lot of G6B, it will activate synthase. And this is the same in the liver as well. The phosphorylase, so this is usually done, the phosphorylase acts to break down glycogen. And whenever we break down glycogen, it's because we have, in the muscles, we have low energy. So whenever we have AMP, AMP usually indicates low energy levels. AMP will activate phosphorylase to break down glycogen and then release all that glucose so the muscle can use it for energy. If there's a lot of ATP, that means the cell is in a high energy state. We're gonna inhibit phosphorylase because we don't want to break down glycogen when we have a lot of energy in the cell. And then G6B also acts on phosphorylase. If we have a lot of G6B, it activates synthase and it inhibits phosphorylase. So the same is kind of the liver is regulated similarly to the muscle. The only difference is glucose in the liver can actually, whenever we have a lot of glucose, it can actually inhibit phosphorylase. So the muscle uses hexokinase to convert from glucose to glucose 6-phosphate. So any free glucose in the muscle is usually converted to glucose 6-phosphate right away. However, the liver has a different isoform of the enzyme. Remember, it uses the enzyme glucokinase instead of hexokinase, and that enzyme has lower affinity for glucose. So we have more free glucose in the liver. And you kind of want the liver to be regulated by glucose because whenever the liver stores glycogen or stores glucose as glycogen or breaks down glycogen to glucose, it does so to regulate blood glucose levels. So you kind of want this glucose to be able to act on phosphorylase because if you have a lot of glucose, you don't want to break down glycogen in the liver. This kind of summarizes what I talked about with why glucose acts on the liver but not in muscle cells. Okay, so those are kind of allosteric ways to regulate glycogen synthase and breakdown. So they can, our synthase and our phosphorylase can be acted on by post-translational modification. So there's two different enzymes. One of them is called glycogen synthase kinase 3, and then we have protein phosphate 1. What these do is the kinase 3 adds 3 phosphates, and the protein phosphatase 1 removes the phosphate. Whenever we don't have phosphate on glycogen synthase, it's called glycogen synthase A, and this is the active form of glycogen synthase. Whenever glycogen synthase kinase adds the three phosphates, the glycogen synthase is inactivated. So we said that G6P can activate synthase. So what G6P can also do is it can bind to the glycogen synthase B, and it makes it a better substrate for protein phosphate 1. So G6P comes in, binds the glycogen synthase B, and then helps it be a better substrate for protein phosphate 1 or PP1, and it allows it to be in the glycogen synthase A form, which is the active form. Okay, so this is glycogen synthase. The same can also happen with the phosphorylase enzyme. So we have a phosphoprotein phosphatase 1 and a phosphorylase B kinase. And what they do is they kind of the same thing. They add phosphates and remove phosphates. The only difference is the phosphorylase is actually activated when it has phosphates. Whenever we remove the phosphates from phosphorylase, we get the less active version of phosphorylase. So if we go back here, glycogen synthase is activated when it doesn't have any phosphatase on it, and it's inactivated when it has the phosphatase on it. The opposite is kind of different with phosphorylase. Whenever it has the phosphate on it, it's activated, and when we remove the phosphates, it's in the inactive form. And then G6P can also play a role in here. So we said that G6P can inhibit phosphorylase, right? So what it can do is it can bind the phosphorylase A, the active form, and it makes it a better substrate for PP1, which allows it to be in a less active form. There's a lot of phosphorylase and phosphoprotein phosphatase, phosphorylase kinase. It's kind of confusing to keep track of everything that's going on. Do you have any questions on that so far, or no? Okay.
Well, actually, one question.
Okay.
But how did you, what resources did you find most helpful when you were taking biochem?
Just the lectures that Shriver gave us were pretty good, and that's how I get all my notes for these. I use
the recordings, but I'm just not sure if that would be helpful to watch or not.
You have the what?
Like the recordings of Shriver's lecture?
Yeah, those will be helpful. I pretty much go over a summarized version of everything she went over, and I use some of the pictures that she has because I think they're really good.
Okay, cool.
To explain.
This is a lot more helpful than reading the Boeing book. So.
Yeah, I mean, hopefully, when I upload this, you can probably slow it down and write everything down to keep track of everything I'm talking about. Okay.
Yeah, I probably will do that.
Okay, so that was we mainly talked about glycogen in the muscle and the liver and how glycogen is used. So glycogen can actually be stored in the brain. It's stored in special cells called astrocytes. Astrocytes are supporting cells for neurons, and basically they store the glucose for neurons. So this over here is astrocyte, and this is a neuron. What they do is they take the G6P right here, and then they can store it as glycogen. And whenever the neurons need energy, they break it down. So the glycogen would be broken down G1P, and then you can convert it to G6P. G6P can enter glycolysis, become pyruvate, and then pyruvate can become lactate with lactate dehydrogenase, and then lactate goes into the neuron, and then the neuron runs lactate to pyruvate, and then pyruvate can enter the TCA cycle and generate energy for the neuron. So whenever the neuron needs energy, the glycogen in the astrocyte will be broken down to pyruvate, then lactate, and then it's transferred to the neuron. Then the neuron can convert that back to pyruvate and uses it for energy. Whenever you're in a high energy state, you kind of run the opposite reaction. But remember, we said we can acetyl-CoA cannot go back to pyruvate. So instead, you have this glutamine-glutamate cycle. Once you make acetyl-CoA, alpha-ketoglutarate can be converted into glutamate, and then glutamate can enter, can leave the neuron, enter the astrocyte, and then glutamate can go into alpha-ketoglutarate, and then it can go to malate. Malate can then go to pyruvate, and then we can store the pyruvate as we do gluconeogenesis, make glucose, and then store it as glycogen. So this is kind of a way to link pretty much everything: the TCA cycle, glycolysis, gluconeogenesis, and glycogen metabolism. And it shows you how the two different cells in the brain work together to regulate energies in the neurons and the astrocyte. Okay, so that's all I have for glycogen. Do you have any questions on any of the glycogen stuff I talked about?
Not yet.
Okay. That's good. All right, so this is the pentose phosphate pathway. So the pentose phosphate pathway, let me zoom in on it. It starts with glucose 6-phosphate. So glucose is converted to glucose 6-phosphate. That can enter glycolysis, or when we have a lot of glucose, some of the glucose 6-phosphate will enter this pathway, which is called pentose phosphate pathway. Glucose 6-phosphate is important because it links a lot of things. So glucose 6-phosphate is involved in glycolysis, so you can run G6P, convert it to fructose 6-phosphate, and you would run your glycolysis. We can also convert glucose 6-phosphate to glucose 1-phosphate, and then we would do glycogen metabolism, which everything we talked about. And then we can also use the glucose 6-phosphate to run in the pentose phosphate pathway, and we'll talk about why the cell would run things in this pathway. So it can also be called the pentose shunt or the hexose monophosphate shunt. Basically, the whole point of why this evolved is when the cell has a lot of glucose, we can't just keep running glycolysis with that glucose right away, because we'll need to, because remember, glycolysis needs NAD for GAPTH to run, and we'll run out of NAD sources to keep running glycolysis. So what we can do is we shunt some of the glucose into the pentose phosphate pathway, and then the pentose phosphate pathway can then be linked to glycolysis, because it makes intermediates that can enter glycolysis. So that was kind of just the general overview of what the pentose phosphate pathway is. So it starts with G6P, and then it connects with glycolysis with two intermediates. So the two intermediates are GAP. So GAP would be that. So this is your first substrate when you enter your payoff stage, or it can enter as fructose 6-phosphate, which is it's kind of later on in glycolysis. So it can enter. So fructose 6-phosphate is after glucose 6-phosphate. The pentose phosphate pathway has two different phases. Okay. One of the phases is the oxidative phase, and the other is the non-oxidative phase. Based on what the cell needs depends on if it runs the oxidative phase or the non-oxidative phase. The first thing you run is the oxidative phase, and then some of the substrates go into the non-oxidative phase. But the way the cell runs these is it's not run linearly. They actually, even though the substrate from oxidative phase go into the non-oxidative phase, the two pathways are run separate from each other. And depending on what the cell needs is what will determine if we run the oxidative or non-oxidative. And this all occurs in the cytosol. Okay, so we have the two phases. So oxidative phase will generate something called NADPH. NADPH is important because it's used. I have the uses of it over here, but it's mainly used as oxidizing a. No, it's used as a reducing agent, and this is used for a lot of things like fat. When we talk about fatty acid synthesis, we'll see why NADPH is important because it's used in all the reaction in fatty acid synthesis. And then the other phase we talked about is the non-oxidative phase. Basically, the non-oxidative phase is just converts between 3, 4, 5, 6, and 7 carbon molecules, and then we end up with glycolytic intermediates, which can then enter glycolysis. Okay, so if we zoom in here on the picture,
Hey Manuel,
yeah.
sorry to interrupt.
No, no, you're good.
I just didn't want to leave on you, but I have that meeting at 5. Are
Okay.
you gonna keep talking and record this, or that I can watch it later?
I might just stop it and then record it later when I have my iPad. Just basically
Okay.
talk over everything again.
Sounds like a plan. I just didn't want to like leave and be like bye.
No, no. I think what I'll do is I'll cut it off to glycogen, and then I'll record pentose phosphate pathway later, and then I'll upload that one. I think
Okay.
that's what I will do.
Awesome. Thank you.
Yep. Thanks for attending.
All right. I will see you next time. This was really helpful. So.
Was it actually or?
Yeah. No. I mean I've learned more biochem right now than I have anywhere else. So.
Okay, that's good. All right.
Cool. Thank you.
Yep.
Bye.
Bye.